The overall goal of this procedure is to determine the thermal properties of leaves in a contact-free manner.
This method can help answer key questions in the field of molecular farming about in-process control of plant growth and health.
The main advantages of this technique are that it is contact-free, fast, and does not damage the sample.
The implications of this technique extend toward automated inspection of plantations with a mobile detector to define feeding and nutrition on a plant-by-plant basis.
To begin plant cultivation, flush mineral wool blocks with 1 to 2 liters of deionized water each. Then, flush each block with 1 liter of a 0.1% fertilizer solution. Place 1 tobacco seed in each block and flush with 250 milliliters of fertilizer solution, ensuring that the seed is not washed away. Cultivate the plants for 7 weeks in a greenhouse or phytotron. Once the plants have grown, either harvest intact, undamaged single leaves for plant characterization or use intact plants. To begin leaf thickness determination, prepare and autoclave 50 milliliters of a 2% mass by volume agarose solution in phosphate buffered saline. Allow the solution to cool to 40 degrees Celsius and then add the solution to a petri dish containing a single leaf sample. Cool the sample at 4 degrees Celsius for 30 minutes to solidify the agarose and embed the leaf. Then, use a vibratome to cut the agarose block into 200 micrometer slices. Mount 5 transversal leaf sections on a glass slide with cyanoacrylate. Use a microscope or dial gauge to determine the thickness of vein-free areas of the leaf sample.